components of lymphatic system, then what are the types of disorders or infections associated with lymphatic system, then what are the various factors that will be influencing lymphatic targeting, then what are the advantages or benefits of going for lymphatic targeting, then the novel targets for lymphatic drug delivery systems, or what are the various carriers that could be used for lymphatic drug delivery. Then very important thing, what are the polymers and ligands that could be used for preparing any type of novel drug delivery systems. And then very important is the key focus areas that you should consider while you are preparing any type of novel drug delivery system. So though I have selected the area of lymphatic system, but I will try that you get a overview that how you can prepare novel drug. Okay, so if we see the components of lymphatic system, you can see in this diagram that there are various components related to lymphatic system like lymph vessels, then central lymphoid tissue constitutes of bone marrow and thymus, then peripheral lymphoid organs are lymph nodes, spleen, epithelio, lymphoid tissues, then circulating pool of lymphocytes. This is very important because when there is any type of infection in these components, then only we have to go for treating those disorders, or they will generate various types of lymphatic disorders. So we will see one by one what is this component and what are the main functions of these components. This is very important because if you know the detail of these components, then only you will be able to target, or you will be able to understand the infections related to these components. Okay. So first is lymph. So what is the important function of lymph? As you all know that it contains various types of nutrients as well as toxins and cellular waste products. And these are transported to and from cellular tissues. Okay. It is very important component of our body. The lymph vessels constitutes lymphatic capillaries, lymphatic vessels, and lymphatic trunks. So the size is very important in these components or in these lymphatic vessels. So when we target or when we prepare novel drug delivery system, we try to prepare systems which will be such a small that they will go and enter in those capillaries or vessels. Okay. So lymphatic capillaries, as you all know, tissue fluid from intestinal space into lymphatic pathway. The lymphatic vessels, they carry lymph from tissue spaces to venous system. The lymphatic trunks, they transfer lymph to two lymphatic ducts, which eventually expel or lymph fluid back to the blood. I know these are very common things that you might be thinking that we all know it already, but it's very important to brush up your knowledge before I go into deeper lymphatic targeting. Okay, fine. The next components include in central lymphoid tissues are bone marrow and thymus. These are very important ones because bone marrow produces all types of potent lymphoid cells which help in differentiation of B lymphocytes. Then thymus, they help in differentiation of T lymphocytes. You all very well know that immune system of our body is very important. And if immune system is disturbed, then various types of disorders you can see. Very common one that you all know is AIDS. In AIDS, what happens is HIV virus, it goes and enters, or it goes and stays into the central lymphoid tissues as well as peripheral lymphoid organs. And peripheral lymphoid organs include lymph nodes, spleen, and epithelial lymphoid tissues. Now, what is very important is when we use any type of conventional dosage forms, then what happens is drug, they don't reach completely to these organs. Okay. Why? Because in our body muted, and when they are distributed into various organs, then very less amount of drug reaches to specific lymphatic tissues or lymphatic system organs. Fine. Then comes lymphocytes. We all know they are mature B and T lymphocytes circulating in blood. Now we will see what are the diseases or disorders related to lymphatic system. You can see here diagram. Here first is AIDS, then lymphadenitis, then lymphadenopathy, lymphangitis. Here you remember that lymphadenitis is lymph node infection. Generally, ITIS means it is any type of inflammation. Okay, when it is associated with any organ, then it is inflammation in that organ. Like here it is lymphadenitis means lymph node infection. Then lymphangitis is inflammation of lymph vessel. Adenopathy is lymph node enlargement. In this, lymph nodes are enlarged. Next, you can see lymphatic filariasis, which is also called elephantiasis. See here, the lymphedema, lymphocytosis. We will in details also.
Excuse me, ma'am.
Yes,
Ma'am, slides are not moving.
slides are not moving.
Yes, ma'am. Okay, ma'am.
Now you can see which slide?
Yes, ma'am. Slide number, I think 8.
6. Okay, this is 7th.
Yes, ma'am. Now it's
Fine.
fine. Yes, ma'am.
Okay, now it is visible?
Ma'am. Yes, ma'am.
Okay, fine. So here you can see lymphoma. What is lymphoma? Cancer. So this is lymphoma means related to lymphatic system. Now there are two types of cancers, Hodgkin's lymphoma and non-Hodgkin's lymphoma. Then this is splenomegaly and this is tonsillitis. Now we will see in details about these diseases and symptoms. Here you see first one is lymphadenopathy. Adenopathy means in this the lymph nodes are enlarged. Okay. And lymphadenitis means inflammation. It is a type of inflammatory condition. Now what happens is why these can occur? It could be because of infection, inflammation or cancer. Infection could be any viral, bacterial or fungal infection. Then it includes strep throat, then mononucleosis, then HIV infection and infected skin wounds. The reasons which can cause lymphadenopathy or lymphadenitis. Then what is lymphedema? Edema, what do you mean by edema? Edema means swelling or accumulation of any type of fluid. So this is called edema. Now when this is in lymph or lymphatic, what happen is it will cause blockage in the lymphatic system. So why it is caused or what are the reasons for it? It is caused by scar tissue from damaged lymph vessels or nodes. Okay. And it is also seen when lymph nodes are removed from those who have or radiation to remove cancer. This is the reason for accumulation of fluid in lymphatic system. The next is lymphoma. As I told, lymphoma means cancer, cancer of lymph nodes. It occurs when lymphocytes multiply uncontrollably. Okay. Next is lymphangitis. So as I told, when it is itis means this is inflammation. So it is inflammation of lymph vessels. Then that occurs means when you are born with this only. It is a type of malformation in the lymphatic system. Then lymphangiomatosis. It is the presence of multiple or widespread lymphatic vascular malformations. Then intestinal lymphangiectasia. This is a condition in which loss of lymph tissue occurs in the small intestine and it leads to loss of protein, then gamma globulins, albumin and lymphocytes. Next is lymphocytosis. This is a condition in which there is higher than normal amount of lymphocytes in the body. Means lymphocyte amount is increased. Then in lymphatic filariasis, it is an infection which is caused by parasite and it causes the lymphatic system not to function correctly. So in these two slides, we discussed various types of infections or disorders which are related to lymphatic system. Now, what we will be seeing is that if you have to target the lymphatic system, then if you are going for any drug delivery system or novel drug delivery system, then what are the factors that will be affecting lymphatic system? So you see, this is a chart in which you can see first is particle size. As I told you, in lymphatic capillaries and in various components of lymphatic system, size is very important. So if you reduce particle size or vesicular size, then those novel drug delivery systems can be taken directly to lymphatic system. Okay. Then charge incorporation. You can incorporate positive charge, negative charge, and these charges have affinity to lymphatic organs. So what happens is, suppose you are preparing for example, if you are taking liposomes, you can prepare neutral liposomes, you can prepare positive or negative liposomes. So liposomes also have natural affinity towards this lymphatic systems. But by modification or by incorporating charges, you can prepare delivery system that will target the drug directly to lymphatic system. Okay. Then surface modification with ligands. This is very important when you are preparing novel drug delivery system. Then you should know various types of polymers and ligands, because these ligands, they help in taking or help in targeting the novel systems directly to the particular organ in which we want the drug to be released. Okay. So there is one more benefit. If the drug is directly going to particular organ, then what happens is, then very less dose you will have to use. Okay. Or very less dose will be required, because it will not be metabolized or degraded. And you all know that if taking any type of antiviral drugs, and those drugs are already, they are very toxic. So it is very important benefit that you are taking any drug and you are preparing a type of novel system which directly goes to the lymphatic system and releases the drug. Okay. Then you can use the coating of polymers in the delivery system. Then lipophilicity is very important. You can use highly lipophilic compounds for targeting lymphatic system. So in charge incorporation, you can use negatively charged surface particles which have very high affinity. The neutrally charged surface particles, they have medium affinity, and positively charged particles have least affinity. Affinity to lymphatic organs. Then we will see a molecular weight. Molecular weight is also very important parameter. So above 1000 Dalton is absorbed by capillaries, and above 16,000 Dalton molecular weight drug is absorbed by the lymphatic capillaries. Now we will see what are the important advantages of lymphatic targeting. See, this is not only advantage of lymphatic targeting, but also this is advantage of novel drug delivery systems. So if we start, we can enhance oral absorption of biomolecules, then we can overcome multi-drug resistance, then we can minimize dosing frequency, we can reduce dose. So by reducing dose, cities will be reduced. Then we can enhance pharmacokinetic parameters. Then we can increase concentration of the drug at target site. Then we can enhance or improve the bioavailability of the drugs, and we can stabilize the drug at gastric pH. So these are important advantages of lymphatic targeting of any drug. Okay. So see, now this is a chart in which we have tried to discuss why novel systems are used or why novel carriers are used. See, when we used to have conventional drugs, then what were the limitations of conventional drugs? Drugs which have poor bioavailability. So what is bioavailability? It is the amount and extent of reaching of drug into the systemic circulation. Okay. So if the drug, very less quantity of the drug reaches to the systemic circulation, means it has poor bioavailability. And if drug has low permeability, short half-life, or extensive metabolism, then we can prepare novel delivery system or novel carriers of those drugs. Okay. And what will be the advantage of novel carriers? Bioavailability will be enhanced, then permeability will be increased. You can improve stability, you can provide sustained release, you can reduce dose frequency. Okay, so if you are preparing controlled release, then you can try, or you will be getting steady state concentration. For that, zero order release will be achieved. Okay, so what we do is we design a delivery system in such a way that predetermined quantity of the drug goes to the predetermined site and releases the drug. So site as well as amount, these two are important factors that have to be considered while preparing novel systems. Now you can see mechanism. This is diagram which is taken from internet only. So here you can see that nanocarriers can act passively or actively. Okay. So solid lipid nanoparticles and nanocapsules, they can reach the target position via passive targeting mechanism. And for active targeting, what we have to do is biochemical interactions should be present, like affinity between receptors or tumor cells and their ligands, or between antigens or tumor cells and their respective antibodies. So here you can see that the nanocarrier is into the lymphillary or into the lymphatic components, and it will release the drug there. So if you release the drug, then what will happen is that the infection, or the, for example, if you go in the case of AIDS, then HIV virus is present. So they will be going, and those drugs will be killing the viral infection or virus. Okay. So you can see here that by passive targeting as well as by active targeting, we can achieve this lymphatic. Now you can see that what are the main important novel targets that could be used for lymphatic drug delivery. This is very important. Like novel techniques, in novel techniques what can be used, and in targeting approaches what can be used. So novel techniques include nano formulations, then surface modifications by various types of ligands. Then you can use biotechnological techniques, then bioinformatics, then nano diagnosis, green synthesis, and phytoactive formulations. And in targeting approaches, you can use carbohydrates, then proteins, polymers, vitamins, antibodies, peptides, and enzymes. See, here this image has been taken from one of our papers. So in this, you can see that enzymes, polymers, vitamins, antibodies, peptides, proteins, and carbohydrates could be taken. Okay. So why I have taken this diagram is to show you what are the examples. Like in the case of carbohydrates, you can see we can use galactose and mannose coating can be done. Then for in the case of protein, you can use lectin, albumin. Then in peptides, like antibodies, IgG. In vitamins, you can use folate. In polymers, you can use polyethylene glycol, polylactic glyceride, then N-isopropyl acrylamide. Then in the case of enzymes, you can use amino acyl tRNA synthetase, then DNA. I'll just increase. DNA topoisomerase 2, then farnesyl transferase, fructose 1,6-biphosphate, aldolase, and glutathione peroxidase. Okay. Fine. Now, for example, I have just one slide. This is related to an infection which is called lymphatic filariasis. So it is, as you all know, that it is a parasitic disease. Okay. And the filarial parasite spreads by mosquito. And what happens is it damages and dysfunctions the lymphatic system. So here, main causative organism is filarial parasite, which is Wucheria bancrofti and Brugia malai. And what are the main symptoms you would be seeing? you would be seeing fever, skin abnormalities, swelling of limbs and genitalia, and hydrocele in male elephantiasis. Then prevention is by insecticide sprays, use of insect repellent creams, and insecticide impregnated bed nets curtain. But for treatment, we can prepare novel systems like solid lipid nanoparticles, okay, and take synthetic as well as herbal drugs, and we can incorporate those drugs into the novel systems, and we can target those systems into lymphatic organs. Okay, so for that, you can prepare novel systems, and in that you will have to do some in vitro in vivo studies such that you get to know that the drug is reaching to the to particular organ or not, and the drug is acting or not, or drug is killing those infections or not, that you have to perform. Okay, basically for targeting or for checking that the drug has been reached or not, we do confocal scanning as well as we go for fluorescence microscopy. Okay, in fluorescence microscopy, what we do is we take a fluorescent marker, like you can take 6 carboxyfluorescent, and you can system. So that delivery system will be administered to animal, then what will happen is that it will be circulated to, or when it reaches to the organs, to the lymphatic organs like spleen, lymph nodes, then you can extract, or you can prepare slides of those, and by using microtome and then by fluorescence microscopy, you can see that the drug has reached to particular site or not. So that is very important because we are trying to target. So that targeting has been achieved or not, that is very important. Then we can also perform various types of pharmacokinetic parameters. By that also we get to know. Okay. Now, see, this slide is also very important because when you are preparing novel drug delivery systems, then you should know what are the important polymers or where the polymers can be used. So it can be used in hydrogels, then for polymer drug conjugates, in microencapsulations, then in adhesive biomaterials. Then you can use polymers in micelles, then polymers in gene therapy, polymers in tissue engineering, then polymers in nanomaterials, and polymers in implants. This is a general slide. Okay. Now, this slide is also very important because nowadays we are trying to move from synthetic to natural polymers. So biopolymers, you should know what are the biopolymers. This is a general slide which I have taken because you should know about various types of biopolymers. So first is hyaluronic acid. This is very important, and it is helpful in skin regeneration, the cartilage regeneration, and cell differentiation. So the students or the faculties, those who are doing research, you should know these types of biopolymers. Then comes gelatin. It is used in trachea engineering and bone engineering. Then chitosan microsphere in cartilage engineering is used. In cartilage engineering, it is used. Then alginates, they are used in vascular engineering. Then collagens, they are used for vascular engineering, then soft tissue repairs. Then capillary engineering, dermis engineering, and cell differentiation. Then collagen HA gel is used in vessel engineering. Then you can use elastin in vessel engineering and fibrin in vessel engineering, as well as fibroblast release. So these are some biopolymers which are widely used nowadays while preparing novel drug delivery systems. Now this, I have just prepared 2 to 3 slides to show how novel targets could be used for lymphatic drug delivery systems. And these are the works which has already been some of the scientists. Okay. So first is chitosan alginate nanoparticles. So you can see here, they have prepared nanoparticles using chitosan and alginate, and the drug used is ivermectin. Okay. Now this is a is prepared for ivermectin so that for a longer duration, slowly the drug is released in the systemic circulation. And this, by preparing this or by using these novel systems, improved microfalaricidal activity was achieved as compared to conventional free drug or as compared to plain ivermectin drug. Okay. Next is folate PEG-CKK2 DTPA carrier. In this, folate is used. This is novel carrier for tumor imaging, diagnosis, and targeting therapy. In this, rapid distribution of conjugation to the lymph nodes was observed. Then we can prepare liposomes. In this, you can use galactose, which is category of carbohydrate. Then in this, enhanced lymph node delivery of hepatitis B surface antigen, silated liposome was observed. Okay, fine. Then another examples are, you can prepare nanoparticles, like here they have used N-isopropyl acrylamide methacrylic acid, MMA. In this, nanoparticles are prepared which are targeted to thoracic lymph node and exert their anti-cancer potential. Then you can also prepare nanostructured lipid carriers, NLCs. They have taken drug tamoxifen, and the biodistribution and in vitro anti-cancer activity of this was found high as compared to plain tamoxifen suspension. Okay. So conventionally, we used to prepare what? We used to prepare plain injections, suspensions, emulsions, and tablets also used to prepare. Then solid lipid nanoparticles. These are prepared for clozapine. In this, it was found that bioavailability was enhanced or increased as compared to clozapine suspension. So these just some of the examples I have quoted. These also I have taken from various scientists which have already done their work. This why I have included is the researchers, those who are performing or those who want to work in the novel systems, they get to know that various types of novel systems are available. Like when we see vesicular systems, then you will have to know that liposomes, transferosomes, ethosomes, lyosomes, phytosomes, they are available. Then in particular systems, you can see here nanoparticles, solid lipid nanoparticles. Then various types of dendrimers are available. Nowadays, nanotubes, carbon nanotubes are there. So you can see various types of systems you can prepare according to your requirement or according to your targeting or the disease which you are targeting. So if you are going for research, what you have to do is, first of all, you see what is the research problem or what you are going to treat. Okay. Then after that, what is the problem in the current existing therapies or methodologies that are already available? Then what will be new, or what will be your efforts in enhancing the knowledge to the current knowledge? That is main, or that is the focus in which you have to go for, in which especially novelty of your work is very important. So novelty as well as applicability. Then these are some of the examples. This is work of my student, PhD scholar Saurabh Srivastav, and what we did was we developed andrographolide loaded solid lipid nanoparticles for lymphatic targeting. Okay, so andrographolide is a natural compound you can see here, and in that we had formulated, and after formulation, in vitro and in vivo evaluation studies were done. So this just I have showed you to that you can develop various types of novel systems for targeting of various organs. Then this is also our paper. In this, mebendazole loaded nanostructured lipid carriers were used. Then this is review. M-Form work in which we had prepared zidovudine loaded surface engineered liposomes. As I told you that in liposomes, you can add various types of charges. So in this, what we did was, first of all, we used mannose coating. Okay, so we prepared mannose coated liposomes, then we prepared positively charged liposomes, negatively charged liposomes. You can see here, mannose coated gave best results, then negatively charged, then positively charged, and then plain liposomes. Okay, so they were prepared, and after preparing, we had done fluorescence microscopy in which we observed that the drug reached there or not. Then this is also work of our student Abhijit Ghosh. He had done in which he had done lymphatic targeting based on novel modified nanosystems. So thank you so much. I just tried to visualize to give you a to visualize you what are novel drug delivery systems and how they are beneficial, or how you can try to prepare these novel delivery systems. And now I'm open for questions. If you want to ask any questions related to preparation or evaluation or development of novel systems, then we can discuss it. Thank you so much. Yes, please. Any questions or queries? See, these are conferences. Why they are organized? They are organized so that if any person is coming from any field or from any related field, then you can interact with them. Interaction part is very important. Okay, so I think it will be good if you all interact, or if you all discuss any problems related to research or development of novel delivery systems. कोई क्वेरी नहीं है? Thank you so much.
Uh, Thank you so much, ma'am, for sharing your deep knowledge with us. It will be a great pleasure for us to listen you again in our future events. Thank you, ma'am. Thank you so much.
Thank you. Thank you so much.
All the participants are requested to please be online. Our second session will be started shortly.
Hello, Disha ma'am. Hello.
Yes,
Good
sir.
afternoon, sir.
Yes.
Good afternoon.
Hello. Good afternoon, sir.
Am I
Disha
audible?
ma'am, you can proceed.
Okay, sir.
Yes, sir. Your voice is clearly audible.
Okay.
Good afternoon, sir. For today's second scientific session, we have with us Dr. Sunil Kumar Jain. I welcome sir on behalf of Columbia Institute of Pharmacy, Raipur, Chhattisgarh. I now take this privilege to introduce our second speaker to the participants. Dr. Jain is presently working as Senior Assistant Professor, Department of Pharmaceutical Sciences, Guru Ghasidas Central University, Koni, Bilaspur. He is PhD and MPharm from Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. He has one book chapter, one completed R&D project, and about 35 publications to his credits. He has about a teaching experience of more than 18 years in university services. With these words, I would like to invite Dr. Jain for his deliberation. Please, sir.
Thank you, Dr. Disha and Dr. Rajnikant. Uh, it's a very good platform to discuss the things that we need to uh, doing research in the infectious disease. Actually, I have
Thank you, sir.
very experience in the. Actually, we have seen many research papers that I have research fields for the non-infectious disease actually for the treatment of cancer as well as the diabetes and H. pylori also. And presently, I am also working on that uh, treatment of some fungal diseases also. So I have some experience regarding the same. Uh, your topic is actually current landscape and novel drug systems for infection global perspective. So I will touch upon some facts and some studies about the infectious diseases and some novel formulations which are need to be uh, used for the treatment. So I am starting to present PPT.
Yes.
Okay. Yeah. So is it visible to you all?
Yes, sir.
Okay.
Please make it full screen, sir.
Yeah. Yeah.
Yeah.
Okay.
Now it is okay.
So okay. I will wish to address some uh, infectious diseases and nano devices uh, used for the treatment of certain infectious diseases. Okay. As we know that there are two types of diseases, infectious disease as well as the non-infectious disease. Infectious diseases are diseases that are caused by pathogen, certain bacteria, fungi, right? Virus involved are that, and that they are contagious. Okay. Similar to that non-infectious diseases, as we know that these are not communicable disease and diseases such certain diabetes, cancer, and so many diseases are there and belongs to that category. So I wish to say that certain pathogens that involves that infectious disease. Now we see the statics about the infectious disease. Mostly peoples from the developing countries are infected with the infectious disease. As it is given as that 14 and 17 million people nearly all live in the developing countries. And these are prone to very much in the children as well as the old age peoples. And those persons are working in the healthcare system. So particularly children, pneumonia, diarrhea, and malaria are leading cause of death among children underage. And these are very infectious diseases are also restricted to the health of adults, causing disability, a poor quality of life, decreased productivity, or even death. Okay, so these persons, those are infected but with one infectious disease, become more susceptible to other diseases. Okay, so we should research also. Some common infectious diseases, as I told you, that viruses, bacteria, fungi, parasites, right? The viruses called common cold, influenza. Recently, we have come across the COVID-19 virus also, stomach flu, hepatitis, right? And bacteria, it is also called strep throat, salmonella, tuberculosis, whooping cough. Okay, ringworms caused by the fungi, giardiasis, toxoplasmosis are caused by the parasites. So there are so many diseases that are infectious, and we require efficient treatment for them. As you know that so many people are very prone to that infectious diseases. Those with suppressed or compromised immune systems, such as those receiving cancer treatments, living with HIV, are on certain medicines. So these medicines treatments also caused reduced immune system, or we can say it is cause our body more susceptible to the diseases. Young children, pregnant women, and adults over 60 are more prone to that infectious diseases. Especially those people are not vaccinated in their childhood. They are also prone to the infectious disease. Healthcare workers and persons traveling to areas where they are more exposed to the mosquitoes, viruses, dengue virus, Zika viruses. So people are at high risk of this infectious diseases. These are some pictures about infectious disease: smallpox, measles, swine flu, herpes, and most of the vaccines are available for the infectious diseases. And research is going on for the developing vaccines for many more infectious diseases coming in future, like chickenpox, tetanus, hepatitis A, hepatitis B, human papillovirus, influenza, malaria, and all these. Polio is already eradicated from India also. So many vaccines are available, and vaccine development is also very much coordinated. We will discuss in the next few slides what are the role of nanotechnology in developing vaccines also. Okay, so these are certain things we need to address. The anti-infective drugs are having certain problems, physicochemical properties, and pharmacokinetic problems are there. Certain problems we need to say that they have the reduced solubility. They have the very narrow window. We need to address. So certain challenges that exist, and they poses certain problems in the treatment. For major example, as I mentioned here, establishment of cellular and anatomical privileged sites, such as the blood-brain barrier and blood-testis barrier. This leads to low-level replication in these compartments, which are inaccessible to conventional. So nanotechnology able to transfer these membranes and are therefore promising tools to be investigated for circumventing this obstacle. So those areas where common conventional drugs will not reach, the nanoparticle technology will give a gate to go and treat the disease at a particular site. Other challenges in the viral application treatment includes the use of RNA interference technology, a popular approach for the treatment of many infectious diseases. So problem is that we have to see the inability of RNA to cross the cell membrane due to large molecular weight an anionic charge, rapid renal clearance, uptake by phagocytes, and toxicity due to stimulated immune response. All present limitations which prohibit their clinical utility. Now, incorporation of siRNA into nanocarriers can also overcome these limitations to achieve successful inhibition of viral replication. Now, we have to discuss what are the different aspects that give an insight about the nanotechnology, and what are the properties the nanopharmaceuticals exhibit for the better treatment for viral diseases. Now, first point, we need to say that small particle size, which can facilitate drug delivery into anatomical precise sites. Second one is the large surface area to volume ratio. And as you know, if you reduce the size, it will provide a more surface area, which ensures that large drug payloads can be accommodated. Tunable surface charge, as we know that we can change the surface charge. It can be, if it is required, positive charge. We can impart positive charge. If it requires negative charge, we can impart negative charge. So there is a tunable surface charge available with the nanopharmaceuticals. Content properties, this result in intrinsic antiviral properties. This silver, if you prepare silver nanoparticles and dendrimers, they also having the carriers also having the properties to treat viral disease. Next point is the possibility of drug encapsulation, functionalization by the formation of stable structures or surface modifications with polymers such as PEG can all leads to optimized drug dosing and improved delivery by increasing stability and drug retention times. It is possible with the nanoparticles that we can encapsulate variety of drugs. Means we can incorporate hydrophilic drugs, hydrophobic drugs, and these modifications, surface modifications, is possible with the certain polymers. So this causes increased stability and drug retention time in our body. Okay, so it is also next point is that engineering. We can do engineered. We can prepare engineered nanoparticles. Okay, so drug can be vastly improved by engineering nanoparticles with targeting moieties to increase specificity to desired cell types, target tissues, or subcellular compartments. We can use certain ligands. We can target it by using certain receptor targeting. We can do so. It is very much useful for the antiviral therapy. Now we can classify this nanocarriers. As we can say, organic nanocarriers are there, and inorganic nanocarriers are there. So organic carriers, we can say polymeric nanoparticles. Both types of things are there. Nanocapsules and nanospheres are there, and liposomes are there. Micelles are there. Dendrimers are there. Solid lipid nanoparticles are there, and in addition to that, lysosomes are there. You can use transfersomes are there. Virosomes are there. So many organic carriers we can use as for nanopharmaceuticals. Inorganic nanocarriers like gold nanoparticles, silver nanoparticles, and we can also use certain metals like titanium, zinc, copper, and prepare them their nanoparticles. These are certain schematics, we can say figures about the nanocapsules, nanospheres, liposomes, micelles, dendrimers, and gold nanoparticles. Okay, so polymeric nanoparticles, as you know, that we can say that particle size in the range of 10 to 1000 nanometer. they are made of macromolecular materials. Means we can use polymers like synthetic polymers, synthetic polymer or polymers from the natural origin. Okay, so based on that, we can get the nano spheres or nanocapsules. Based on the method of preparation, we can get. And also, all these three parameters, we can also modify surface charge also by taking different parameters. Okay, so because of their small size, what is the mechanism of nanoparticles? You can see by the slides. Because of their small size, very small size, nano size particles are there. So how they have worked in our body, how they have become used as a targeted list. Okay, so as we can see, first step is there. Nanoparticle carries the pharmaceutical agent inside its core, while its cell is functionalized with a binding agents. So you can see that this nanoparticle containing this pharmaceutical agent, or you can say drug, inside its core, okay, and while its cell is functionalized with a binding agents. Through this binding agents, second step we can say, through this binding agent, the targeted nanoparticle recognize the target cells. A slight nanoparticle cell interact with the cell membrane. Now there is a start with the interaction with the cell membrane. Now this cell membrane, this nanoparticles is ingested inside the cell and interact with the biomolecules inside the cell. Now we can say this is the engulfing of that part. The next point is the nanoparticle break down, maybe by the erosion, by the dissolved, and maybe drug is released by a diffusion mechanism. So this is the mechanism of taking off of target regulatory of this nanoparticles. This small size of polymer nanoparticles can facilitate capillary penetration uptake by cells, resulting in increased concentration at target sites. As I told you that surface modification is possible with certain hydrophilic polymers to reduce non-specific interaction with the serum proteins, decrease susceptibility to opsonization, and to defer uptake by phagocytosis, thereby prolonging the drug half-life and further altering the biodistribution of the target profile of the drug. Surface modification is possible with certain polymers. This helps for better targeting also. Now, one example I have took from some research papers, use of nanocapsules. This limited antiviral distribution could maybe due to the permeability glycoprotein efflux transporter. Problem is that, problem kya hai? What is the problem? Limited antiviral distribution to brain tissue, maybe due to the permeability glycoprotein efflux transporter. And it is found that solitol HS15 is an excipient that is able to inhibit PGP, thereby improving drug distribution across the blood-brain barrier. Now, research is done. This excipient is used, loaded with the HIV protease inhibitor, that is the indinavir, so significantly increased uptake in the brain and testes of mice compared to control mice when only indinavir solution was administered. So when this excipient is used and this excipient is loaded with the HIV protease inhibitors, that gives more better results as compared to the only indinavir solution. Now you can see different technologies, different dosage forms. Some are in the market, some are in the clinical trial. Okay, so you can see preclinical and clinical nanotechnologies for treatment of HIV infection. HIV infection technologies are there, injectable nanoparticles, injectable lipid nanoparticles, oral nanoparticles. So many drugs are utilized for developing certain nanoparticles. Motivation are there, common modification, the reduced dose frequency and target macrophages. Okay, so these are the testing conditions. We can say the treatment for TB, tuberculosis. Different drug regimens, combination of drug regimens are used, and technology we can use that oral lipid nanoparticles, nebulized nanoparticles, and aerosolized micro or nanoparticles. So these are also in the some preclinical stages. Now we're talking about the treatment of malaria. So injectable immune liposomes, microemulsion, injectable nanoparticles, injectable nanoliposomes are developed, and these are in the testing conditions. Now, as I told you in earlier slide, that so many vaccines are available for the infectious diseases. So, what are the role of nanotechnology in developing of vaccines? So, nanoparticles play a very important role. They serve as a adjuvant for the vaccine delivery. Now we can see that antigens are either adsorbed onto their surface or entrapped in the matrix of polymethyl methacrylate. Nanoparticles. See that these nanoparticles containing the influenza antigen may protect against disease to a greater extent than the antigen alone. If you see that if you use as a antigen alone, it will not protect the person for longer period of time. If you is there, if you particles in influenza antigens may give the prolonged effect. Okay. Okay. By decreasing the particle size and increasing the hydrophobicity of these particles, it improves the adjuvancy, and the particles offers a prolonged and controlled presentation of the antigen in the immune system. Now, that is about the nanoparticles. Liposomes are also giving so many options, so many opportunities for the drugs which are used for the infectious disease. There are certain advantages or certain properties which gives better treatment when you prepare as a liposomes. So as we see, the advantage of liposomes. These liposomes provide selective passive targeting to RS organs, increased efficacy and therapeutic index, increased stability of encapsulated drug, reduction in toxicity of the encapsulated agent, site avoidance effect. You can only target the drug to the targeted tissues, not to the non-target tissues. Only need to target where it is desired. Okay, improved pharmacokinetic effects means reduced elimination, increased circulation times, and flexibility to couple with site-specific ligands to achieve active targeting. One more point is you can encapsulate variety of drugs. Means you can encapsulate solid particles. You can encapsulate liquid drugs. You can encapsulate hydrophilic drugs. You can encapsulate hydrophobic drugs. So liposomes encapsulate variety of drugs, and it is also gives an opportunity to tag with the different ligands for selective targeting to different organs. Now there is a mechanism which are involved in the liposomes. mechanisms that cell liposome interaction, localized drug effects, enhanced drug uptake, molecules with wide range of solubility and molecular weight can be accommodated, flexibility in structural characteristics. Now, this is the mechanism of work cell liposome interactions. Liposomes can include water-soluble drugs in the aqueous space and lipid-soluble drugs within their membrane. The core part contains, if it is we want to encapsulate drug, water-soluble drug, it should be in the core part. And if we want to drug, it should be within the membrane, core part. Okay, so they will interact with the cells in 4 ways: absorption, endocytosis, lipid exchange, and fusion. The absorption first, it will adsorb on the surface of the cell. Endocytosis, cell engulfing cell will be taken up by the liposome will be taken up by the cell. Lipid exchange, the exchange of lipid molecules between a vesicle bilayer in water and nanolayer. And fusion, a process of joining 2 or more things together to form a single entity. Now, second point is that localized effects. Liposomes help the drug within selected sites or selected cell types. This is because of their large size and low degree of penetration of epithelial and connective tissue barrier, which tends to remain at the site. Enhanced drug uptake, it can occur by the vesicle cell fusion or by endocytosis. Molecules with wide range of solubility and molecular weight can be accommodated, and flexibility in the structural characteristics that I discussed earlier. Now, as I have discussed that we can use nanoparticles as a carrier for the vaccines. Similar to that, liposomes also offers certain characteristics so that it can be used as a carrier for vaccines. First point is that a non-immunogenic substances may be converted into immunogenic one. We can convert non-immunogenic substances to immunogenic. Hydrophobic antigens may be reconstituted. A small amount of antigen may be suitable as immunogens. Multiple antigens may be incorporated into the single liposomes. Adjuvants may be incorporated into antigens into the liposomes. Longer duration of functional antibody activity may be achieved. You can get the longer duration of functional antibody activity. Toxicity and allergic reactions of inclusion in soluble synthetic antigens may be presented as membrane-associated antigens in insoluble liposomal matrix. So, following points will give an idea that liposomes is a very good carrier for vaccine delivery. Now, one example is that liposomal diphtheria toxide, and we can see the charge over the surface, how it will give the response. Liposomal diphtheria toxide, both neutrally charged. See the things that both neutral and negatively charged, they're equally responsible to produce the same immune response. Their positive charge liposomes produce reduced response. So charge will play a very important role. Okay. So in this, we have to understood what should be the charge and which charge is suitable for the better response. We can see this liposome as a carrier of antigens, diphtheria toxide, cholera toxins, herpes simplex virus type 1 antigen, and so many. It's a very old study actually, and so many nowadays, so many antigens can be delivered via liposomes, and liposomes can be used as an effective carrier for the so many antigens. Now, liposomes are also used for topical delivery, not only liposomes. We can say transformosomes, nisomes show very good potential for a topical drug delivery. I am working on that the topical drug delivery and using of certain drugs related to antifungal therapy. And I have one problem also that some agents like iconazole, fluconazole, uh, having the limited solubility that poses many problems for the delivery of drugs. So use of lipid nanoparticles or nano NLC, nanolipid constructs can give better topical applications of certain antifungal agents. No doubt, we can also increase the solubility of certain drugs, antifungal drugs, by using by certain solubility enhancement techniques also. We can use liposomes for pulmonary delivery, as we discussed earlier in the treatment of HIV and TB and malaria. That liposomes can be used as the aerosolized delivery or the pulmonary delivery or various anti-infective agents. Now, leishmaniasis, I wish to discuss this leishmaniasis. This actually what is the role of liposomes and nisomes. They play a very important role for the treatment of this leishmaniasis. As we know that it's a parasitic disease caused by infection with the leishmania parasite, which are spread by the bite of flavotomine scent fleas. Okay, and its treatment and what is called actually it inhibits cells are liver and spleen, and treatment so many antimonial agents are used. Antimonial agents can damage the heart, liver, and kidney. So this is the very much very side effect of the drug that this antimonial drugs, if you used for the leishmaniasis, it causes great damage to our body. So if you encapsulate such drugs in liposomes, not only greatly reduce the dose needed to treat the infection, but also deposit the conjugates at infected Kupffer cells. Here, at least some of the liposomes are digested by the parasites. So we can use liposomes here very efficiently. We can also use the, as I told you, nisomes. We can also use the transformosomes for the treatment of leishmaniasis very efficiently. Now, if you see that amphotericin B liposomes, we have come across during this period of coronavirus, and one more disease happens is black fungus, and so many persons administer this amphotericin B liposomal formulation. So, if you see this amphotericin B liposomal formulation, and because of their properties, it gives better therapeutic response as compared to the plain amphotericin B. As you discussed, antibiotic is there. This ambisone, it is marketed product is the ambisone. It is a liposome formulation of amphotericin. Diameter is, or we can say size of that, 50 to 100 nanometers. Works to decrease toxicity related to amphotericin B. And what are the, as I told you that different side effects are there. This called actually renal toxicity. Very much cause the renal toxicity, very harmful to the body. So if you use it by then incorporating to the liposome formulation, it gives better results. Now these are certain list of liposomal drugs already in the market for the treatment of fungal infections, protozoal infections, malignant lymphomatous meningitis, HIV-related Kaposi, sarcoma, hepatitis A, influenza, analgesia, menopausal therapy. So many liposomal formulations are available now, and so many companies are developing liposomal formulations. Now, some inorganic nanoparticles we can also use. Most effective metallic nanoparticles against bacteria, virus, and other eukaryotic microorganisms, particularly due to the inherent inability and bactericidal potential of silver. As I told you earlier, that this different metals we can use. These having the dual effects. So silver also gives the bactericidal effects. Other mechanism, what is that? The release of silver ions, which enhances antimicrobial activity, cell membrane disruption, DNA damage. Various others, as I told you, titanium, zinc, and copper, iron oxide, titanium dioxide, we can use, and they have demonstrated specific antiviral activities. Combined thing that so many nanomedicines which are approved under evaluation are in the market for the treatment of viral infections. So many virosomes, liposomes, inactivated virosomes, pegylated interferon, this virosome vaccine, and therapeutic vaccine, solid lipid nanoparticles, wet lipid nanoparticles are under development. So important thing is that challenges, what are the challenges with the nanotechnology? You see the challenges. Challenges is that most important the drug resistance. If you administer the drug in large amount, it causes the drug resistance. First point, this needs to be addressed properly. That what is the continual therapy, continual administration of drugs cause the drug resistance development. So if you use the nanotechnology, definitely it will reduce the drug and clear the problem of the drug resistance. So you can use the drugs very properly. Okay. First point is that reducing the cost, definitely the cost will be more if you develop new drugs and also it will be nanotechnology based products. Because and if development countries will not be able to cope up with that condition. So solution may be that we can use the existing drugs and formulate it as a nanocarriers. Existing drugs, cost of the existing drug is very less, so you can reduce the cost. But if you use the newer drugs, the cost of the itself and drug moiety is very high. And if you administer develop as a nanocarrier, the cost of the whole form. So reformulating existing drug in nanocarrier is one opportunity. Now, if you see that example, as I told you, MB-SON, amphotericin B liposomal formulation. This is not effective, and it is very costly. Okay, excipients used in nanosystems such as lyoprotectants can increase the cost of many treatments. So, treatment preparation cost, development cost is very much more. High techniques, skilled persons are required, highly sophisticated instruments are required for production of nanotechnology, nano formulations. The cost is a very much concern and major challenge towards development of this nano formulations. Other is second point is the synthetic synthesis and storage conditions of nanoparticles may not be conducive to conditions in low resource countries. Researchers will need to enable the reproducibility in bulk production while considering the environmental effect of the nanosystem. So if you see that all these things, if you develop a bulk formulation, it will not work in all conditions. Okay. So what is the what we need to do it with three-dimensional construct of multiple components with preferred spatial elements. Subtle changes in process or composition can adversely affect the complex composition of nanomaterials. So we need to face environmental conditions also, storage conditions, while developing a nanoformulation. Regional and national differences within and between regulatory authorities, especially when running multi-center international trials, will be a challenge during the clinical trial stage. So, conditions, clinical trial studies are very much difficult, and it's a major challenge. Finally, efficient acceptability of these nanosystems will need to be addressed and will need to be discussed properly. Future research will explore the possibility of multifunctionalization to achieve concurrent drug delivery and imaging by a fluorescent signal, for example, to determine in vitro localization and specific cell tissue compartment targeting. Using targeting ligands like peptides and proteins are molecular recognition strategies, and multiplexing in order to increase the spectrum of disease that can be treated in heterogeneous population by simple, reliable, and cost-effective methods. So this is the future. Not only nanotechnology is future, it is present also. Now, I wish to conclude with some certain points. Nanoparticles have very special properties that make them attractive for nanomedicine. Nanoparticles can be made of lipids or polymers, then decompose once a target is released and deliver a pharmaceutical agent. So these are, we can say, biodegradable in nature. Nanoparticles can be functionalized with antibodies to target their binding towards cells. Nanoparticles can be used in diagnosis through the detection of biomarkers. They can be engineered to incorporate conventional antiviral properties with those modifications that are unique to nanosystems, like ultra small and controllable size, large surface area to volume ratio, and ability to tailor the surface with the possibility of multifunctionalization. Now, this targeted nano technology will give an idea to treat infectious disease wisely. Now, I wish to conclude and thank you all with wish na sarveshantu dharamaya sarve bhadrani vishantu ma kashyatu bhagaved. Thank you all.
Thank you so much, sir, for sharing your deep knowledge with us. It will be a great pleasure for us to listen you in again in our future events. All the participants are now requested to kindly participate in queries section. Is there any question from participants?
Good afternoon, ma'am. Hello.
Good afternoon. Yes.
Good afternoon, sir. So my name is Arushi Sarupi. I have one question. So what are biomimetic particles?
Biomimetic particles, we can actually, you can use two types. Bio means having the same properties, means having, which are the part of the body. You can see using liposomes, you can certain phospholipids. Okay, certain things which are part of our body. So these are biomimetic. If you, one simple example, I will tell you that what happens if you administer parenterally in our body any components, any nanoparticles, or anything, it will serve as a foreign body. Okay, if you use the part of our body, जैसे HSA है, human serum hemoglobin, है ना, या phospholipids हैं, these are parts of our body. So body will accept properly. Okay, so it will help to more targeting, better targeting. Yeah. Any more questions? But I'm not happy with all these things. Actually, I wish to discuss all the matters physically. Actually, online presentation is not very much.
Yes, sir. Just we are planning to organize a different seminar
Yeah,
offline. Next time, international conference will be there.
yeah, yeah.
So we are happy with you, sir, that you are so much interested to talk on this particular topic. And definitely, very soon we are planning to organize that, sir. An offline conference that will be started very soon. And this is,
If
I
the,
think, last conference from Columbia Institute of Pharmacy, which is going
yeah,
to be held online.
yeah, yeah. Because the online mode is not good, and if you participants are not front of you, then it's very difficult to understand and deliver them. Yeah.
Yes.
Thank you. Yeah. Thank you, Dr.
Thank
Manik, for providing me an opportunity to discuss and
thank-
interact with.
thank you, sir. You're most welcome, sir. Always you're
Yeah.
most welcome, sir.
Yeah.
Thank you, sir.
Thank you.
Thank you, sir. All the participants are requested to please be online on Google Meet. We are very soon we are starting our third scientific session with Dr. Vijayalakshmi Ghosh.
Audience, just please be online. Speakers are just ready. Ma'am, this ma'am, you can share screen
Okay, sir. Okay, sir.
by opening her slide. It's a very interesting topic that will be given by Dr. Vijayalakshmi ma'am.
Is it visible, sir?
Yes, it's visible. Participants from PG courses, MPharm, pharmacology, MPharm, basically specialization from pharmaceutics. Okay. They will be benefited by this topic, nano-emulsion formulation for topical anesthesia.
Mr. Chhatrapal Sahu, please mute off your mic.
This ma'am. Madam has come.
Yes, sir. Just a minute.
You can ask to ma'am if she will suggest you to stop screen from your side.
Am I
Ma'am,
visible?
shall we start?
Yes,
Yes, ma'am,
are
you
my
are
slides
visible.
visible to you?
Yes, ma'am. Yes, ma'am. It's
Okay.
visible. For third scientific session, we have with us Dr. Vijayalakshmi Ghosh. I welcome ma'am on behalf of Columbia Institute of Pharmacy, Raipur, Chhattisgarh. Welcome, ma'am. I now take this proud privilege to introduce our speaker to our participants. Dr. Ghosh is presently working as an assistant professor in GD Rungta and Technology, Bhilai, Chhattisgarh. She has done her PhD in nanobiotechnology and masters in biotechnology from VIT University, Vellore, Tamil Nadu. She has completed her postdoctoral research from Paris, France in 2015. She has one patent, 8 book chapters, and 13 national and international publications to her credits. With these words, I invite Dr. Ghosh for her deliberation. Please, ma'am.
Thank you, ma'am, for the introduction. And I also thank Columbia.
Hello.
Yes, am I audible? Hello.
Yes. Yes, madam. You are right.
Okay. So thank you, Columbia Institute of Pharmacy.
Ma'am,
Yes,
there is some disturbance with your voice. Means
there is.
some extra. There's some disturbance, ma'am, along with your
Ma'am,
voice.
Is it
some
okay?
background voice that is coming this side.
Yes.
There is no one here. I am alone.
Maybe network issue. Ma'am, please, you can keep continue. After some time, it will again it will be solved.
Okay. So good afternoon, everyone. I am Dr. Vijayalakshmi Ghosh, working as an assistant professor in biotechnology in GD Rungta College of Science and Technology, Bhilai, Chhattisgarh, India. So today I'll be talking on nano-emulsion formulation for topical anesthesia. So before, this is my outline of my presentation. So we need nano-emulsion, then formulation of nano-emulsion, followed by the characterization of nanoemulsion. Then there is a brief introduction about local anesthetics. Then the local anesthetics that typically the lidocaine-based formulations, and characterization and stability analysis of the lidocaine-based formulations. Finally, the diffusion kinetics and cytotoxicity assay. So before knowing about nanoemulsion, I just want to introduce what is emulsion. So emulsion is the suspension of two immiscible liquids, and here we use emulsifier to stabilize the emulsion, which lowers the interfacial tension between the surface, surface of the two immiscible liquid. And based on the dispersed phase, the emulsion, it's divided into two types: oil in water, that's oil droplets are dispersed in water, and in water in oil emulsion, water droplets are dispersed in oil. So what are nanoemulsions? Nanoemulsions are metastable submicron dispersions with droplet diameter in the range of 20 to 200 nanometers. So due to this low droplet size, they are also called mini emulsions or submicron emulsions and ultrafine emulsions. So again, this ultra low droplet size, it provides potential advantages of this nanoemulsion over the conventional emulsions, like high physical stability, high bioavailability, low turbidity, and all these potential advantages, they make this nanoemulsion system attractive systems for application in food, cosmetic, and pharmaceutical industry. So as they serve as delivery agents of lipophilic bioactive compounds, such as drug in case of pharmaceutical industry, for delivery of flavor and antimicrobial agents in food industry, then solubilization of water-insoluble pesticides in agrochemical industry, and finally, as vehicle of lipophilic skincare and personal products in cosmetic industry. So as we discussed, these are the advantages of nanoemulsion. So the nanoemulsion, they provide better stability to particle aggregation and gravitational separation due to their droplets. And this low droplet size, they scatter the light waves very weakly. That is why the system is, instead of the conventional emulsion which looks turbid in color, the nanoemulsions, they are optically transparent or translucent. And these nanoemulsions, they can be designed to have novel rheological properties. That means we can have high viscosity and gel-like characteristics by adding the gellifying agents. And also, these nanoemulsions, they increase the bioavailability of the lipophilic bioactive compounds which are encapsulated in the nanoemulsion. So how we prepare nanoemulsion? So basically they are prepared either by high energy method where we provide energy, and in low energy method where we provide very less amount of energy. So the low energy methods include spontaneous emulsification using magnetic stirrer or vortex, membrane emulsification, solvent displacement, and phase inversion composition. And the high energy methods of nanoemulsion preparation, that includes the use of ultrasound generators, that's the sonicators, and high pressure homogenizer where we provide high pressure, and then also we have microfluidizer. So once we have prepared this nanoemulsion, where we are going to use. So these are some of the applications where the nanoemulsions can be used apart from the drug delivery. So in case of drug delivery, we can use the pharmaceutical bioactives. And
Excuse me, ma'am.
yes.
Ma'am, slides are not moving.
Okay.
Yes, ma'am. Now it's fine, ma'am.
Then can you share the slides? I have already provided the slides. Is
Okay,
it
ma'am.
moving?
Yes, ma'am. It's slide number 7.
Yes.
Ma'am.
Slide number 7, applications of nanoemulsion.
Yes, ma'am. Yes, ma'am.
Okay. So the applications of nanoemulsion, it includes larvicidal agent that's as nanopesticide, and also it can be used for delivery of topical products such as antimicrobial agents, and also in food industry, it can be used for in the beverage products to enhance the property. And can you see the 8th slide?
Yes, ma'am. It's visible.
Okay. So before starting the topical anesthetic formulations, I want to give a brief introduction to the local anesthetics. So what are local anesthetics? They provide anesthesia only locally, unlike the general anesthetics. And most of these local anesthetics, they are topical anesthesia. They are used for topical anesthesia. And when we talk about these local anesthetics, they are composed of 3 molecules, or the structure can be divided into 3 parts. The first part is a lipophilic part, which is aromatic, and the second part is a hydrophilic part, which is a amine end. And there is a link between this lipophilic and hydrophilic end, that's between the aromatic and the amine end. And based on this link, which is provided between the aromatic ring and the terminal amine, we can divide the local anesthetics into 2 types. So they can be either aminoamides, and they can be amino esters. Some examples of aminoamides are lidocaine, prilocaine, and some examples of amino esters are benzocaine and tetracaine. So here is a table, as you can see. It's based on the use of the local anesthetics starting from the year 1884. As we can see, in the 1884, or in the beginning, the commonly used local anesthetics were having the linkage as ester linkage. So the examples are cocaine, benzocaine, procaine, and tetracaine. From the year starting from 1884, 1900, 1905, and 1941. But starting from 1947, there has been introduction of amino amides local anesthetics, which include lidocaine, mepivacaine, bupivacaine, and ropivacaine. So we can say that the commonly used local anesthetics these days are mostly amino amides. Okay. How these local anesthetics work? What they do? They block the nerve conduction by reversibly binding with the D4S6 part of the alpha subunit of voltage gated sodium channels in the nerve, and that's how they block the transmission of pain. So basically, they block the sodium gated channel in the neurons, and hence they prevent depolarization, and they block the transmission of pain. So we don't feel any pain once we have applied the local anesthetics. So these are some of the examples of formulations of local anesthetics or the topical anesthetics. First one to discuss is EMLA, that is eutectic mixture of lidocaine and prilocaine. And there is LMX, it's a liposomal cream of lidocaine, which has lidocaine 4%. And then there is Versatis, which is a patch formulation having lidocaine 5%. So here of these three formulations, the commonly used formulation in USA and Europe is EMLA. And the EMLA, it caused methemoglobinemia, which resulted because the conversion of hemoglobin to toxic methemoglobin, which is induced by the prilocaine, which is present in EMLA. So as you see in the EMLA, along with lidocaine, we also have prilocaine. So lidocaine and prilocaine both are present as 2.5% of the total formulation as the compounds. Okay. So to avoid this or to overcome this side effect of prilocaine, what we have studied that I am going to explain in the future slides, which was being carried out in the University Paris Descartes in Paris, France. So we tried to formulate a new formulation which has lidocaine, but it did not have prilocaine, which caused the side effect in the newborns. So to introduce the formulation, it's the eutectic mixture of lidocaine and fatty acid, and prilocaine, it is replaced by fatty acid, and prilocaine, it was present in the earlier version of EMLA. And the formulation which we prepared, it had less number of excipients, and we followed a very simple protocol, and the formulations were thermodynamically stable. So first, we formulated a eutectic mixture. So what is a eutectic mixture? Eutectic mixture is the specific composition of at least two solid components that produces a change of phase to liquid at a certain temperature. And this temperature is called as eutectic point temperature. And as we are talking about emulsion here, so both the solids which we have taken, both are lipophilic in nature. So first one is your lidocaine, and second one is the fatty acid, and we have taken here or as the fatty acid. So as you see, the melting point of lidocaine is 68 degree, and the melting point of lauric acid is 43 degree, 43.2 degree. But when we do lipophilic solids, the eutectic mixture, the melting point was 5 degrees centigrade. So as you see, the liquid, the phase, and at this eutectic, this mixture was liquid at this 5 degrees centigrade. So this is known as eutectic mixture. So after formulating the eutectic mixture, then we proceeded to prepare the nanoemulsion. So the first one, the eutectic mixture formulation by lidocaine and lauric acid. Then this eutectic mixture was vertexed with other excipients. We're going to talk in the next slides what are the excipients we have taken. But then After adding the excipients to the organic phase by mixing this other excipients with the eutectic mixture of lidocaine and lauric acid, and it was vortexed for 1 minute. After that, water was added to the organic phase. By vortexing, we prepared the oil-in-water nanoemulsion. So you can see the process is very simple and it's a low energy method. We just vortex the formulation, the excipients along with the eutectic mixture, and then finally added water to prepare nanoemulsion. So, what are the excipients we have taken? We have used emulsifiers, that's polysorbate 80, polysorbate 85, polysorbate 20, PEG 400, holoxamar F68, and also there was some concentration of olive oil used, and unsaturated fatty acid, oleic acid was used, gelling agent glycerol was used. So, all these excipients were optimized for the formulation. So, out of total 124 formulations which we have screened, I am showing you 7 best formulations, and that's 10B, 10W, 11K, 12Q, 12S, 13I and 13J. Again, of these 7 best formulations, we have the lowest droplet size. It was obtained with 12S formulation, where we have taken lidocaine 3% and eutectic phase is 6%. That means other 3% is the fatty acid. And the formulation, it was visually translucent in color, and it was a viscous liquid, unlike gel, because we see it has very low percent of lidocaine and the fatty acid. And the size which we obtained is 144 nanometer, and polydispersity index or PDI is 0.09. As you see, it's a very low PDI. That means the formulation is highly stable with a zeta potential value of -64 and pH of 7.1. This is the graph showing the 12S formulation of the result as taken by the droplet size analyzer or the nanosizer. So here the average droplet size is 144 nanometer and polydispersity index is 0.09. And this is the transmission electron microscopy image of 12S formulation. So this is, we can see the droplets were spherically in shape, and also it provided additional data about the droplet size, that droplets are between 100 to 200 nanometer size. Then we studied the rheology, we checked the viscosity, and we compared the viscosity between 12S and 10W. These are our best formulations. So 12S, it was viscous liquid and it was translucent and it had low viscosity compared to 10W, which is a gel, and it was transparent in color and it had high viscosity. Then we moved on to check the stability of the formulations. Here again, as we said, 12S and 11K are our best formulations, so they were stable. That means the droplet size did not change with time. But the formulation like 10W and 12Q, they were unstable because as we see, the droplet size increased as we increased the storage time. After checking kinetic stability, that's the by keeping at the room temperature, we also checked the thermodynamic stability of our formulations by centrifuging at 10,000 RPM for 30 minutes, then by subjecting it to heating cycle, that's 4 degrees centigrade and 40 degrees centigrade and 4 degrees centigrade alternatively for 3 cycles. Then by freeze and thaw states, by keeping at -25 degrees centigrade and 25 degrees centigrade alternately for 3 years. So here in the cycles, in either in case of heating cooling or freeze thaw, so it was kept for 24 hours in each temperature per cycle. So basically it took 6 days to complete one study. Both the studies were completed in 12 days. And also thermodynamicity was confirmed by subjecting it to heat sterilization. So simply by autoclaving at 121 degrees centigrade for 15 minutes. So to this thermodynamic stress, 12S and 11K formulation, they were stable. There was no change in droplet size and there was no physical separation of the constituent phases. Then these best formulations, based on their size and stability, they were checked for diffusion kinetics to skin. So for checking the diffusion kinetics, we have used Franz diffusion cell. So as we see, this cell, it has two chambers. The upper one is donor chamber and the lower one is receptor chamber. So this donor chamber and the receptor chamber, they are separated by a membrane. So this membrane either can be kept as a skin explant or a synthetic membrane. So we put our compound on the donor chamber and then through this membrane it passes through the receptor chamber. Then from the sampling port we take sample and we see how much our compound has diffused to the membrane. For this study we have used as membrane skin explants and synthetic membranes which are silicone based. When we talk about silicone explants, skin explants, these are obtained after plastic surgery. As we see in France, in Europe, it's plastic surgery is very common. So we have obtained consent from the, we don't say patients, but because they are healthy volunteers. So from the volunteers, we have taken the sample after their skin plastic is over. Then we have used that as membrane between our donor chamber and receptor chamber. So here we see the diffusion kinetic study when we talk about the effect of drug concentration. So if you have same drug concentration between two formulations in 10B and 10W, both of them had 6% lidocaine. But based on the excipients, we say if in the organic phase, the number of excipients, not the number, but the composition, the percentage of excipients is more, then the diffusion kinetics is slower. And if there are less number of excipients, so if the drug concentration is more in the organic phase, the diffusion kinetics is faster. This is according to Fick's law. So here, as we see, 10W had less number of excipients, so the lidocaine percent was 50% in the organic phase. So the diffusion kinetics was faster than 10B, which was 25% of the drug in the organic phase. So it was slower diffusion kinetics. Then, when we compared our formulation with the commercially available formulations, we found that our formulations, the best formulation 12S, it was having the highest diffusion rate than 10W, followed by EMLA and LMX, which are the commercially available formulations. So again, we can say which the products or the formulations prepared by us, they had better size, better stability, and better diffusion kinetics. So we have better properties, that is good. But before releasing it into market or before applying for patent, we should also check its cytotoxicity. So as we are applying this formulation as topical anesthesia on the skin, so we check the cytotoxicity in the skin cells. As all of us know, skin has more number of keratinocytes. That's the 90 to 95% of the epidermis is keratinocytes. So we conducted our cytotoxicity studies on keratinocytes and also fibroblasts, which also constitute some part of the skin cells. So here, as I discussed, we obtained the skin explants from the volunteers after their plastic surgery. So first we removed the fat layer. Then this skin explants were prepared by adding them into 0.25% trypsin EDTA pucks buffer to separate the epidermis from dermis and to obtain the keratinocytes. Then the separation of the epidermis layer and keratinocytes were obtained. And these keratinocytes were studied for cytotoxicity study. So cytotoxicity study on X plants were studied for 15 minutes, 30 minutes, 45 minutes, 60 minutes, then 2 hours and 4 hours. And there was no significant difference was observed until 4 hours. So we can say our formulation, which we are going to patent or file a patent as anesthetics, it had no toxicity up to 4 hours. Then also we check the cytotoxicity on the cell lines. So there are keratinocytes. The keratinocytes from NCTC cell line and Hackett cell line were used for studying cytotoxicity. There are two assays, neutral red assay and MTT assay, which were used. All of us know that neutral red uptake assay, it provides a quantitative estimation on the number of viable cells, because this neutral red, it binds to the supravital dye, that's the lysosomes of our cell, and it provides the data on the viability. And as the cells begin to die, the cells, they are unable to incorporate neutral red in their lysosomes. So as the cell viability decreases, so neutral red uptake also decreases. So this can be used for studying viability assay. And there is another assay which is MTT. It is a colorimetric assay for assessing cell viability. So in this case, NADPH dependent cellular oxidoreductase, this enzyme, what it does, it reduces tetrazoleum dye, which is MTT, to its insoluble formazole. And this insoluble formazole is purple in color. And then what we do, we solubilize this insoluble purple formazole by adding dimethyl sulfoxide, and then we take the absorbance at 550 nanometer. And this absorbance value, it reduces with loss of viability. So again, it is used for viability assay. So you see, we have taken MTT assay in case of NCTC cell line. So there is less absorbance. You can see A, B, C, D, E. So these three are, the line A is the formulation, it's the control cells. Then B, C, D, E, they are our formulation. And F, G, H, they are commercial formulations. So we can see the commercial formulations, they are almost faint in color. That means there is cell death is occurring. In our formulations, there is less death, or the cells are living alive. So viability in case of our cells, it is high. So here we see, even in our formulation, the 12Q, 12R, they are little toxic because the lidocaine percentage is higher, that's 6%. So there is some cell death along with it, which is a commercial formulation. But our best formulations, which are 12S and 11K, you see there is no death. So similar again, NCTC, after incubating for 2 hours, we found EMLA is toxic. And MTT for 30 minutes, 12Q, 12R, EMLA and LMX, they are toxic. NCTC, MTT test for 2 hours, again 11K, 12W, 10W, 12Q, 12R, EMLA, LMX, they are toxic. But our 12S formulation, it is the best. It is not showing toxicity in any of the tests. Yes. Okay. So also we have performed the cell line study by using Hackett cell line, the keratinocytes. Similar results were obtained. 12Q, 12R, EMLA, it is toxic, but our formulations, they are not toxic. The 12S and 10W, which are best. Again, similar results. So we also conducted some study on fibroblasts. So here, again, the best formulations are not toxic. 12R is toxic due to high concentration of lidocaine. And here EMLA is toxic. Fibroblast 57, again 12Q, 12 EMLA, LMX, they are toxic. 12S, 11K is not toxic. Yes. So similar results were obtained in CHO 38 fibroblasts also. So in all the cases, as we see, there is the best formulation, which is 12S. There is no cytotoxicity report. So here we conclude that emulsions were formulated using eutectic oil of lidocaine and fatty acid by replacing toxic prilocaine as in EMLA. And the best formulation, the optimized one, which is 12S, it had the lowest droplet size of 144 nanometer and lowest PDI value of the polydispersity index of 0.09, and it is also highly stable. The 12S formulation, it also had better diffusion kinetics than other formulation and also better diffusion kinetics than the commercial formulation, which are EMLA and LMX. In the cytotoxicity studies on keratinocytes and skin explants, it demonstrated that 12S and 12G are toxic, 11K and 10W are slightly toxic, whereas 12Q, 12R, EMLA, LMX, they are highly toxic. So we suggest that this 12S formulation, it can be possibly used as topical formulation for local result of our experiment. The outcome is European patent. So we have filed this patent and it has also been granted the year 2018. These are some of my references. I would like to acknowledge Rungta group of institutions for providing me the facility to work, and I also like to thank University Holiday Park in some CNRS where I have conducted my studies, and also I would like to thank Columbia Institute of Pharmacy for inviting me to deliver this lecture. Thank you. If you
Thank
have
you,
any
ma'am.
questions, please.
Thank you, ma'am, for sharing your profound knowledge with us. Now, the queries are invited from participant side. Queries and questions are invited from participant side. Is there any question? Is there any question to anyone? Any doubt? Okay, ma'am, I don't think there is any question. Thank you so much, ma'am. Thank you. Thank you so much, ma'am. So thank you all the participants for joining us. Tomorrow we will start our scientific session